Information on Wednesday, but mostly Wednesday and Friday I'm going to be doing a little bit of review. Obviously I can't review the entire semester in a day and a half. So for the final will be 50% the material since the last exam and 50% cumulative. Okay. Now the 50% that's cumulative is going to be mostly stuff that relates to the stuff since the last exam. Okay. So you may have to know some amino acids and how that relates to forming a peptide bond. Things like that. But I'll give you some more examples of that stuff. So although it's cumulative, you don't need to focus on everything since the beginning of the semester. Most of it will be on the last part since the last exam. So transcription, translation, replication, urea cycle, amino acid metabolism, and regulation. Okay, so we're going to continue on regulation just to remind you what we talked about last week. We talked about negative regulation, and that negative regulation, no matter what the outcome, is the regulation that involves repressors. Again, we're talking about bacteria. Okay, eukaryotes is a little bit different. And by a little bit, I mean a whole lot different. So the repressor will either bind to the DNA and shut down transcription. Okay, so transcription is normally on. We're making messenger RNA. Repressor binds to a signaling molecule of some sort. Then it binds to the DNA, shutting off transcription. Alternatively, a signal can cause a repressor to disassociate from the DNA. So the repressor is normally bound to the promoter, transcription is off, some molecule comes and binds to it, it falls off, makes a messenger RNA. So this would be the example of the lac repressor, right? The lac repressor is normally bound to the DNA. If lactose, allolactose is around, it binds to the repressor, falls off, and transcription can occur. Okay. This is an example of the trip operon, and we'll talk about that today. Then we have positive regulation, and positive regulation involves activators. And in bacteria, this is enhancing the activity of RNA polymerase. So the binding of RNA polymerase to the promoters. Binding sites for activators are usually near the promoters because RNA polymerase weakly binds to these promoters, so they need activators around to help them bind to the DNA. It may remain bound until a molecular signal signals it to not be bound. Okay, so here's the activator helping bring RNA polymerase. So RNA polymerase binds to the promoter. What's the function of the promoter? It's the binding site for RNA polymerase. Activators bind to an activator binding site that's usually close to the promoter. A molecular signal comes, the activator no longer is bound to the DNA. Alternatively, the activator may only bind in the presence of a signal. So here's the RNA polymerase, doesn't really bind really well. A molecular signal comes in, binds to the activator brings the RNA polymerase in better connection with the promoter. So this would be an example of CAP and CRP that we talked about. So cyclic AMP would be the molecular signal, CRP or CAP would be the activator. They bind together, they can then bind to the region near the promoter and help RNA polymerase bind. So let's talk again about the lac operon. Just to remind you, the lac operon is responsible for making the proteins required for the bacteria to utilize lactose as an energy source. So there are proteins involved in breaking down lactose. 3 genes that what we call the structural genes of the lac operon. That's LAC-Z, that encodes for beta-galactosidase, LAC-Y that encodes for the permease, and LAC-A that encodes the transacetylase. So the permease allows lactose to come into the cell. Beta-galactosidase has 2 functions, converts lactose to allolactose, as well as cleaves it into galactose and glucose. Okay. So these are all important for making sure we can use glucose as an energy source. Again, we don't know what transacetylase does. We think it has something to do with waste. So the LAC repressor is encoded by the gene LAC-I. So just to remind you guys, lowercase italics, that's the gene. Uppercase, no italics, that's the protein. Okay. So the genes, lowercase italics, uppercase, that's the protein. So LAC-I encodes the repressor, the LAC repressor, which is LAC-I. The gene LAC-I has its own promoter, so the promoter for LAC-I. It's independent of the transcription of the structural genes. Okay? So LAC-I has its own promoter, is able to make the repressor protein separate from the ZYA genes. Bless you. The repressor binds to the DNA and the LAC operon on 3 different operator sites, but mostly to the first 2. So remember, here is the LAC operon, here's the promoter for LAC-I, the repressor, our operator sites, LAC-ZYA. The DNA where the LAC repressor binds has these palindromic sequences, so they're the same forward and back. And the LAC-I mainly binds to the operator 3 and the operator 1, making that lovely little stem loop structure. So in essence, the LAC repressor is blocking the promoter from RNA polymerase binding. So that's how it's blocking transcription, is by physically blocking the promoter location so RNA polymerase can't bind. Okay. Again, binding to the DNA so it loops out in the middle. So it binds to the 2 promoters, the DNA is looped out between the 2 sequences. Okay. Now, although the promoter is mostly hidden from RNA polymerase, there is still a little bit of transcription that occur even when the LAC repressor is binding, but not a whole lot. Okay. We just assume that the promoter is blocked enough that very little transcription is actually happening. Okay. So it's about 17 to 22 base pairs of contact on each side of the DNA, again with this palindromic sequence. So the LAC repressor will remain bound to the DNA until it becomes in the presence of allolactose. And again, allolactose is converted from lactose using beta-galactosidase. So that enzyme is converting lactose to allolactose. So allolactose serves as our inducer that binds to the repressor and allows it to disassociate from the DNA. So I'm going to go way back to that first slide we had.
As the inducer to the repressor, or the...
So LAC is normally bound. LAC repressor is normally bound. Our molecular signal in this case is allolactose.
Right.
It binds to the repressor, causes a conformational change, it no longer can bind to the operator. Promoter's exposed, RNA polymerase can now do its thing. Yeah.
That way, in the presence of lactose, it will start moving along.
Right, because those are the genes for breaking down lactose, so they only want to be made if there's lactose to be broken down.
Okay. Get this time.
See, after the 6th or 7th time, it'll get in there.
I'll get it.
Okay, so allolactose, which we call the inducer, binds to the repressor, causes it to disassociate from the operator. And the job of beta-galactosidase is to convert lactose to allolactose. So we can just assume when lactose levels are high, allolactose levels are high, even though allolactose is the molecule that's responsible for binding to the repressor. So in addition to the regulation done by the LAC repressor, the LAC operon is also under the control of catabolite repression. And catabolite repression in bacteria inhibits them from using any other carbon source over glucose. If there's glucose around, you use glucose. It is the best energy source out there, and we don't want to use any other carbon source if we have glucose. So when glucose is present, the lactose genes are turned off. Well, really turned way down. So the catabolite repression is mediated by cyclic AMP and the cyclic AMP receptor protein, or the catabolite activator protein, depending on your book, CRP or CAP. Okay. So catabolite repression is mediated by those two molecules, so cyclic AMP and the CRP protein. When glucose is absent, cyclic AMP goes up, so decrease glucose, increase cyclic AMP. Cyclic AMP binds to CRP, then CRP, cyclic AMP, can bind to the lac operon, turning up transcription. So low glucose, high cyclic AMP binds to CRP, CRP then can activate the lac operon. But that can only happen if there's lactose around. So there also has to be lactose binding to the lac repressor, which is no longer bound to the operon. So it doesn't matter how much we're starving to death because we have no glucose. If we have no lactose, those genes aren't going to turn on either. We got to find something else to eat. Hopefully there's something else around. Okay, so lactose is absent, operon is off. Okay. Doesn't matter what happens if glucose is high or low. If there's no lactose around, it's going to be turned off. The lac repressor is bound to the DNA. No transcription of the operon, even if there's CRP and cyclic AMP. This ensures that the lac operon is only on if there's lactose around. So here's the repressor, no lactose around, still bound to the DNA. Doesn't matter if glucose is high or glucose is low, no cyclic AMP CRP bound, yes, cyclic AMP CRP bound. That repressor's on, it's not going to transcribe those genes. So that's why we call the repressor the on-off switch. No lactose around, it's going to be either be on or off. It's going to be off with no lactose, on if lactose. Now we swim into some lactose. Now it depends on what glucose is doing, if we have glucose around. So we swam into lactose, so the repressor disassociated, but it's only on a little bit. So here's our allyl lactose binding to our repressor. It can no longer make that funky little stem loop. We make a little bit of messenger RNA. It's on a little bit. We run out of glucose. Cyclic AMP and CRP bind. Now we make lots and lots of messenger RNA. We turn up the dimmer. Okay. So just remember, lactose absent, doesn't matter what glucose is doing, it's off. Lactose present, now it depends on, the level depends on whether we have glucose around. Lots of glucose, very little transcription. Run out of glucose, whole lot of transcription. And these things ensure that two things happen. We only turn the lac operon if there's lactose around. And if there's both glucose and lactose, we use glucose first as our energy source. It's a better energy source than lactose. Yeah?
Why do we need lactose present and not just use glucose like by itself?
Because these genes right here, they have nothing to do with breaking down glucose. So we don't need them on if we just have glucose. We only need them around if we have lactose.
So glucose can still be
Mhm.
made as energy even if the repressor's on?
Mm-hmm. Yes, because what What do you need to break down glucose? This is another good connection to the question. What are the genes involved in breaking down glucose? What's the first enzyme you need for breaking down glucose? Hexokinase. The gene for hexokinase needs to be turned on, right? Either one of those, none of those genes are hexokinase. Yeah. Yeah, so all the genes for breaking down glucose have nothing to do with these three genes. Yeah.
So the order of importance for that is going to be lactose, cyclic AMP, and glucose doesn't really matter because it's going to be inversely proportionate to cyclic AMP.
Right. Yes, this is inversely proportioned. So if we have high glucose, this will be low. So high glucose, low cyclic AMP, that doesn't happen. So we have low expression. It's low expression, right? That's this case right here.
Right, thank you.
Yeah, so the levels of cyclic AMP are inversely proportional to the levels of glucose. Yeah.
Okay, so what if you're lactose intolerant?
Okay, bacteria are not lactose intolerant.
Oh, okay.
So only you humans us are lactose intolerant. And actually, I don't even want to relate it. They're not related at all.
Okay.
The only reason that they're kind of sort of related is that if you can't utilize lactose, your E. coli sure can. And one of the byproducts of utilizing lactose is lots of gas. And that's why you're really gassy when you're lactose intolerant, because your E. coli is eating it for you, because you can't utilize it. But basically, you can't utilize the lactose if you're lactose intolerant, but E. coli can't be lactose intolerant. So overall, lactose is low, repressor is bound, no transcription. Lactose is high, repressor disassociates, transcription is permitted. If glucose is high, CRP is not bound, little bit of transcription. We run out of glucose, glucose becomes low, cyclic AMP is high, CRP is bound, lots and lots of transcription. More questions about the lac operon before we move on to the trip operon. Okay, now stick all of your lactose knowledge into a little box and put the lid on it. And we're going to talk about breaking down of tryptophan. Even though it's happening both in E. coli, these are very different mechanisms for regulating gene expression. So just like us, E. coli has to make some of its amino acids. Actually, bacteria make all of their amino acids. And one of the amino acids that they make is tryptophan. So they have a set of genes called the trip operon and the trip operon that are responsible for making tryptophan. Transcription is regulated by a signal we call the attenuator. And the attenuator is a sequence on the messenger RNA that we'll talk about that is responsible for terminating transcription. This does not happen in eukaryotes because, as I mentioned last time, bacteria do transcription and translation at the same time. So the only way this works is if you do transcription and translation at the same time. Eukaryotes, we don't do that. Okay. So this is a mechanism that happens only in bacteria. So the attenuator is a region of the messenger RNA near the 5' ends. And what it does, it makes the ribosome stall. So here's the DNA of our bacteria. Okay, RNA polymerase is going to come in there, and it's going to make messenger RNA. As soon as that messenger RNA is made, and it's made 5' to 3', so the 5' end is going to jump out first. In bacteria, the ribosome jumps right on and starts making protein. Okay. So as RNA polymerase is going, the The ribosome can jump on and start making its protein. That all happens at the same time. I have a much better picture of that than I can draw. Okay, so let's first start talking about the trip operon. So what are the components of the trip operon? Let's ignore the trip repressor right now and just look at the DNA. So here's the DNA. Here is the promoter. And here are our structural genes. So to make tryptophan in E. coli, you need 1, 2, 3, 4, 5 genes. E, D, C, P, A. Yes, they're backwards. So this is the biosynthetic pathway for tryptophan in bacteria. Each one of those genes, E, D, C, A, and B, are responsible for a different step in this pathway. That's all I wanted you to get from that slide. So each one of these 5 genes is responsible for converting into tryptophan. So these genes are responsible, bless you, for making tryptophan. Yes?
Where does corazine come from?
It's another precursor in the cell. You don't need to worry about it. Just realize that these are responsible for making that conversion pathway. Okay, so in that pathway for making these genes, these are supposed to represent each one of the components. So this is supposed to be trip E, trip D, trip C, trip B, and trip A. And B and A actually come together to make one enzyme. Just realize that the messenger RNA made from this DNA encodes each one of these genes, each one of these proteins. So the leader region is 162 nucleotides at the beginning of that messenger RNA. So here's the messenger RNA made from this piece of DNA. The leader sequence is at the 5' end of that messenger RNA. It includes 4 segments that we've numbered 1 through 4. And those 4 segments can make different stem loop structures. So remember, RNA can make these wonderful stem loops, or like our tRNA. It can form these wonderful stem loop structures. Those 4 regions can form some different stem loop structures. If segments 3 and 4 base pair, they form this lovely hairpin structure like this, which we call the attenuator. So the attenuator is actually this structure of the messenger RNA. If segments 2 and 3 base pair, it makes this other structure, and there is no attenuation. Okay. So that leader region can form 2 different structures, either the 3-4 stem loop or the 2-3 loop. Okay. We call the 3-4 the attenuator. We call the 2-3 non-attenuator. So here's the messenger RNA. Here is the leader sequence right here. So here's 1, 2, 3, and 4. And then here is the start of trip E. So again, if you look back at this picture, this would be the leader sequence, and here's where trip E starts. So this is again the messenger RNA. Within the leader sequence, I want you to notice in segment 1, there's 2 codons coding for tryptophan. And those are important for making this work. So here's the DNA. See a prettier picture than I did. Here is the RNA polymerase, and it's making the messenger RNA. Here's the ribosome that jumped on, and here is the polypeptide chain that it's starting to transcribe. So it started going along segment 1, and here it's going to move on to segment 2. Here's 3 and 4 making the attenuator structure. So we're living in a world of high tryptophan. We've got plenty of tryptophan in the cell, tryptophan to go around. If we've got plenty of tryptophan around, that means we have plenty of tRNAs that are charged with tryptophan. So we've got a bunch of tRNAs around. Okay. So if there's plenty of tryptophan, and there's plenty of tRNAs for tryptophan, then we don't need to make any more tryptophan. Right? These genes are responsible for making tryptophan. We got plenty of tryptophan, we don't need to make any more. So we want to stop this from happening. So the ribosome is jumping along, and remember I said there's 2 trip codons right there. So the ribosome gets to those 2 trip codons and says, I need a tRNA for trip. Well, there's plenty around, so it easily finds one and it keeps chugging along. No problem. So it doesn't stop, just keeps going. Okay. The ribosome remains close behind RNA polymerase. They're almost going together. And because it covers up segment 2, so it goes on to segment 2 cannot pair with segment 3. So remember that 2,3 base pair, the segment 2 with segment 3, it can't make this structure because segment 2 is all covered up by the ribosome. So that leaves segment 3 to form the hairpin structure with segment 3. So 3 and 4 form this lovely structure we call the attenuator, and that terminates transcription. Okay. So plenty of tryptophan, ribosome fills those 2 trip codons, no problem, chugs on along as fast as possible. The 3-4 stem loop structure forms, terminates transcription. So this stops. Because we have plenty of tryptophan around, we don't need to make any more. Now we start running out. We started getting a decrease of tryptophan. Now we're no longer making plenty of tRNAs because we're trying to make them, but there's no tryptophan around. So there is no tRNAs or very little charged tRNAs with tryptophan. So now the ribosome gets to those 2 trip codons and says, I need a tRNA for tryptophan, except there's none around. So it stalls. It's waiting for a tRNA to fill that spot. Now if it stalls on 1, 2 is free and clear to base pair with segment 3, so it forms that 2-3 base pairing. 2-3 base pairing means transcription doesn't stop. Okay. RNA polymerase continues, transcription continues, and we make more tryptophan. So if the tryptophan tRNA is scarce, the ribosome stalls on those 2 trip codons. The 2-3 base pairs form, and transcription and translation proceed. Yeah?
I'm getting confused.
Yes.
Which one stalls? Transcription or translation for what?
So the ribosome is stalling.
Okay.
Yeah.
Thank you.
So in bacteria, once this ribosome would have gotten past, another one would have come on. But if that ribosome is stuck there,
It
it's
doesn't have
stalling.
all that junk attached to another one.
And this will continue to keep going. Yeah.
And what part of the 4 areas is there the TRP and the TRP together?
It's in the 1.
It's in the 1.
That's what those 2 little brackets are supposed to be. Those are the 2 trip codons. Yeah.
So then how does that affect the binding of the 3 and the 4 together?
So it's actually either affecting the 2 or not. So if it doesn't stop right here, then it goes and moves on to 2. So this is what it's trying to show you, that it's moving on to 2. If 2 is covered up by the ribosome, then 3 and 4 base pair with each other. Okay. If the ribosome gets stuck on 1, 2 is able to base pair with 3. instead of 3 base pairing with 4. So it's either the ribosome has moved on and covered up 2, so it's no longer available, or the ribosome stalls and is stuck on 1, so 2 is available to base pair with 3.
But do you still need the tRNA of the TRP, both of them, to continue with the 2, 3,
Yeah.
2 as well?
So
But it's just that the
yeah,
surrounding.
so it's like there's a lag in the translation, right? Until we've made enough tryptophan to catch up, and enough tryptophan to charge the tRNAs to catch up. Yeah.
So the RNA polymerase is always going to be continuing the mRNA strand regardless of the level of tryptophan. It's just the translation itself is gonna throttle.
In reality, the stalling actually, the moving forward actually helps it disassociate, but you don't need to worry about that. You can just think of it as it just keeps going.
Okay.
It's much more complicated than just this, but just think about it, the simplistic view of it. Other amino acids do this similar thing, where it puts codons in that leader sequence for whatever amino acid you're trying to make, like leucine or phenylalanine or histidine. So this is a way that we can use the availability of the tRNAs to signal to the cell the availability of the amino acid. In addition to that regulation, the trip operon also has a repressor. So in the presence of tryptophan, the trip repressor binds to DNA. When trip is abundant, it binds to the repressor binds to the operator, and it slows down expression. That's another mechanism to prevent the operon from being on if there's plenty of tryptophan around. So bacteria are using 2 different mechanisms for the same thing, to make sure the operon is on when you need tryptophan and off when you don't. So the trip repressor, let me go back to the picture real quick and I'll come back to that. I didn't put another picture on there. Okay, so this is the trip repressor. So the trip repressor, TRPR, binds to tryptophan. There's plenty of tryptophan around. It binds to the operator sequence, turns off transcription. Okay. So the trip repressor, just like the LAC repressor, is a DNA binding protein, and it binds to DNA. And we can look at the sequence of proteins to determine if they potentially bind to DNA. And one of the sequences we look for is a motif called a helix-turn-helix. And that helix-turn-helix, we can look at it and say, that probably interacts with DNA. And it fits perfectly, if you look at the size, into the major groove of the DNA. So it's a very common motif. So if we see that motif in proteins, especially new proteins we discover, we say, hey, that's probably a DNA binding protein. It's about 20 amino acids, and it includes a helix, an alpha helix, a turn, a beta turn, and then a helix, another alpha helix. So that's why it's called helix-turn-helix. So that sequence is able to bind in specific contacts between the major groove of the DNA and those helixes of the helix-turn-helix. Okay. So the one I'm actually going to show you is for the LAC repressor, but it has a similar kind of motif in most DNA binding proteins. So remember that LAC repressor, how they drew it, kind of looked like that crab structure? Well, each one of those little claws is a helix-turn-helix. So this is the helix-turn-helix, helix-turn-helix, helix-turn-helix. And it fits into the major groove within the DNA. There's other structures that we also look for to bind DNA, like leucine zippers, things like that. But this is just one good example of a motif we can look at to say, hey, this is a DNA binding protein. So another concept that kind of went back to the beginning when we talked about amino acids that make helixes if you know that a set of amino acids make a helix turn helix, and we look at that sequence, then we can say, hey, that's probably a DNA binding protein. So we can determine from that sequence the potential function of that protein. Any questions about the trip operon? So different mechanisms, because one is to break down lactose, the other is to make tryptophan. Yeah.
Okay, so you had mentioned that bacteria could do this with all the amino acids that they
Mhm.
could make their own.
Mhm.
Is this the same process for all the different amino acids?
In general, yes. They have that a leader sequence where they have more than one codon coding for that specific amino acid. The ribosome can stall. They're all very similar to that. They make a little bit different structures. You don't need to worry about that, but they'll make different structures in the messenger RNA.
So why is it important to know tryptophan out of all the amino
That's because it's just the most well studied. We just know the most about it. That's what tends to be that we learn about them because we know the most about them. Especially E. coli, because we always know the most about E. coli. And we're going to just talk a little bit briefly about eukaryotic gene regulation. Now we need to step out of our world of bacteria and think about eukaryotes. So what are our big differences about prokaryotes and eukaryotes? Well, we just talked about one. In bacteria, translation and transcription are coupled. They happen at the same time. In eukaryotes, we have this pesky nucleus problem. Okay. So transcription and translation happen in two different spots. So we can't do that wonderful thing like attenuation in eukaryotes. It doesn't work because we do transcription, translation in different spaces and times. Another thing, in bacteria we have that single, in most cases, circular chromosome. It's just a double-stranded piece of DNA, not really compacted down. It's nice and free and open. Where in eukaryotes, we've got chromosomes, right? Very tightly condensed, large pieces of DNA and chromosomes. I guess I should put them in the nucleus, right? Okay. So another problem. So those two facts change how we have to regulate DNA transcription and translation in eukaryotes. So, like when we talked about with bacteria, a lot of our regulation was based on the interaction of the RNA polymerase with the promoter. Where in eukaryotes, it's a little bit different than that. A lot of our regulation is based on the access to the DNA because it is so compacted down into these very tight chromosome structures. So our regulation requires us to remodel that DNA. So positive regulation is mostly what we're seeing in eukaryotes. So this negative regulation stuff, not so much. Mostly everything we see in eukaryotes is positive. And even a little bit of positive regulation is required for any sort of gene expression. And obviously it's more complicated. Requires a lot more proteins. So just to remind you about chromosome structure. So again, our nicely compacted down DNA. So our DNA is first wound on histones. So here is the histones. They come together making this nice nucleosome and then form into ultimately the chromatin fiber that is wound down into the compact chromatin. Okay. So the DNA in bacteria just looks like this. It's nice and open, it's not all compacted down like this because it's much smaller. But in eukaryotes, we got to get all the way past these structures to even get access to the DNA. Okay, so it's all compacted down. So in essence, our chromosomes are always being negatively regulated because none of the RNA polymerase or anything can get to the DNA. It's always off. So even to get a little bit of transcription, we gotta unwind all of this stuff to even get to the sequence of the DNA. So just to remind you some terminology, the euchromatin is the less condensed chromatin, and it is transcriptionally active, where the inactive is the heterochromatin. Okay, so euchromatin is gonna be the less dense, more looking like this stuff than the heterochromatin, which is all nice and compact. And that's an artifact of being able to see it underneath the microscope. So we have to remodel the chromatin, we have to unwind the DNA from that compact structure by repositioning those nucleosomes, so moving those nucleosomes around. We can modify or use different histones, and then we can just covalently modify the nucleosomes. Okay, so we can either move them around, use histone variants, or covalently modify the nucleosomes. And all of the goal of this is to remodel that chromatin so we can get access to the DNA. Because transcription ain't gonna happen if we don't have access to the DNA. So we'll first talk about the restructuring of the nucleosome. And we call this the SWIF complex, which is these, it stands for the switch sucrose non-fermentable. Don't worry about that, just if you wanted to know what it's called. So this is remodeling chromatin to irregularly space the nucleosomes. So normally the nucleosomes are in very ordered fashion. We're wrapping our DNA around the histones, the nucleosomes are forming in a very ordered fashion. Now we're going to remodel them so that, in essence, we're pulling them away from segments of the DNA. That then stimulates the binding of different transcription factors, and we can allow transcription to start. This requires the ATP to remodel this. So here is our lovely compacted DNA on the nucleosomes, and we are going to expose segments of DNA. You don't need to be too worried about each one of them. I was just giving you some examples of how we expose the DNA. So we pull them apart, we slide one to the side, but in essence, the goal is to expose that DNA. Because if it's all lined up like this, the mechanism for RNA polymerase to start transcription is not going to happen. Histone modification can also alter transcription. So the histones allow recruitment of the enzymes and the transcription factors, so RNA polymerase, all those lovely transcription factors, to come in there and bind to the DNA. So for example, if we methylate a couple of the lysine residues of histone 3 and an arginine of histone 3 and 4, this results in activation of transcription. So we modify those histones, we add some methyl groups to some different amino acids, transcription is activated. It recruits two different enzymes. One of them is histone acetyltransferase that then acetylates certain lysines. That acetylation of lysines results in a decreased affinity for the histones of DNA. What does that mean? It means the histones don't want to bind to the DNA anymore. So the methylation leads to an acetylation. The acetylation leads to the histone not binding well to the DNA. Therefore, it's exposed, transcription can happen. It's reversed by a histone deacetylase that helps to remove those acetylanes. Yeah?
This is what makes the urochromatin?
Yes.
So that's that little space right there, that's
Mhm.
urochromatin.
Yeah.
And what's the other thing that you said? Start up.
The nucleosome modification.
No, but you said like another word besides urochromatin.
Heterochromatin?
Heterochromatin is the compact one.
Compact.
Yeah.
Okay.
So heterochromatin is compact, euchromatin is open and exposed. And we just called it that because that's what we saw on the microscope. We didn't know what was happening. We just saw one that was compact and one that was not. So that's why we named it that.
So in comparison, all this would be like helicase and like bacteria.
It's a little bit different because we're not actually unwinding the DNA strands. We're just unwinding the, so think of the DNA, this is the two strands together, then it's being wound up really, really tightly. So this is going from this to a segment of DNA that we can actually expose. We still have to pull the two strands apart, but we couldn't even get to the DNA if it was like compacted up like this. So this is getting from way compacted to just portions where we can get the different enzymes in there to start transcribing. This is just not a problem we have in bacteria. They don't compact their DNA, so. So that methylation, again, of a particular amino acids of our histones. I should go back to that picture. Sorry. So, let's see if this shows them. So these are the histone complexes in here. Histone H1 is the one that's linking the nucleosome together. So these are modifications of these histone proteins, these kind of green globby ones right here, that are leading to changes in the DNA.